And I thought maybe this brine shrimp puppet could introduce Eric. Would that be too inappropriate or not
No,
professional?
I think that's delightful.
So Eric Dunham, he is a PhD candidate at Montana State University, expecting to graduate this summer. In his thesis work, he investigates how the microbial communities that live under glaciers can survive using hydrogen gas and bedrock materials. At Great Salt Lake, Eric has studied the formation of the mineral dolomite by microbial mats on the lake floor and is currently working on estimating the primary productivity of the lake's photosynthetic cyanobacterial communities. Take it away, Eric.
All right, thanks, Jamie, or thanks to your brine shrimp friend. I'm just getting to here. Can everybody see this?
Eric Dunham started. Oh yeah, there you go.
Yep.
Okay. So as Melody kind of touched on, I want to talk about a newer project we have going on at the lake that is it's in progress right now, and so I thought what I would do is just kind of walk you through what we are attempting to do with this project, how we do it, and have some. I'm going to end with some sort of wild extrapolation of our existing data, just kind of for fun. So, Melody's talk was really about the microbialites being the basis for the food webs at Great Salt Lake. And let's see, let's see if my slides will advance here. Here we go. So, we know that this entire Great Salt Lake ecosystem is really supported by microbial primary production at its base. We have, these benthic microbialite communities that feed the brine flies, and we have the planktonic microbes that feed the brine shrimp. Correct me if I got that mixed up. But and the brine flies and the brine shrimp are a critical food source for all the birds that are supported in this. It's a massive wetland habitat, basically. And one of the things that's sort of strange about this is that nobody's ever really estimated, nobody's ever tried to quantify the primary production at Great Salt Lake. And so that's what we're trying to do with this project we have underway now. So primary production at the most basic level is turning CO2 into biomass. I have this chemical equation up here. We have CO2 and water. And with the input of light energy, photosynthetic organisms can turn CO2 and water into sugar and oxygen. Some of that sugar and oxygen they burn to generate energy, just like we do. And then some of that sugar gets shunted into biosynthetic pathways and gets turned into biomass. So that this biomass is generated through photosynthesis is what feeds brine flies, brine shrimp, and those in turn feed the birds of the lake. And so this, photosynthesis is really the foundation of this ecosystem. And so we want to look for a way to quantify the amount of photosynthesis going on in the lake so we can estimate, how much biomass is being generated by the lake each year. So the way we do this is with a microcosm-based approach. And we have these tiny glass vials. They're called serum bottles, and we'll fill them with lake water, which contains microorganisms. And the CO2 that's captured in the headspace of those vials is turned into biomass via photosynthesis. And since all of this is captured in a little bottle, we can hopefully quantify this process after a set duration of time. So I have a photo here of this is me collecting some water to put into our microcosms. Here's Dr. Boyd filling a few of them on our first sampling trip last July. And the way we actually quantify this is we use 14C. So it's a radioactive isotope of carbon. And if we add a known amount of radioactive CO2 to each of these microcosms, that gets incorporated into biomass, just like a normal 12C non-radioactive carbon molecule would, or carbon atom would. And we end up with radioactive biomass. So we can filter out the cyanobacterial biomass from these vials after we've run our experiment. And we can quantify the amount of radioactivity that got turned into biomass. And then we can estimate from there, how much of the total, how much carbon was turned into biomass during the time period under which our experiment was run. So we can use this data, we can use this radioactivity data. We basically look at the ratio of, how much 14C radioactive carbon did we put into these microcosms in the first place, and what percentage of that was incorporated into biomass, and that's how we can get an estimate of how much biomass was generated during our experiments. So the way this works is we first have to go out to the lake. Here's a picture of Libby Foams, who's another member of the Boyd Lab. We go out in the lake, we just collect water and we start filling bottles. And here on the right side, you can see her filling. We have somewhere between 50 and 100 microcosms that we fill on each sample trip. And the first step in this, like I said, is just to fill the bottle with some lake water that contains planktonic cyanobacteria that are fixing carbon in the lake. So after we've filled our microcosms with lake water, next we need to kill half of them basically. And we'll use this to control for any abiotic reactions that may occur in any non-biological ways in which this 14C we're adding could be captured within our filtered microcosm water, giving us a false signal. So what we'll do is we'll boil half of our water samples into jet boil. This should kill any microbial cells in our water samples, and these heat-killed microcosms should be fixing, quote unquote, fixing less CO2 than our biological control or our biological sample microcosms. So once we've killed half of our microcosms, we next have to find out, how much of this carbon fixation is due to photosynthesis versus other microbial pathways. There are several microbial pathways by which CO2 can be fixed into biomass, and they're not all light dependent. So what we'll do is we'll wrap half of our microcosm bottles in aluminum foil to keep it dark inside, so that as we incubate these during our experiments, we can get a difference between the amount of CO2 fixed in our light exposed microcosms versus our dark microcosms, and we'll be able to use that difference to estimate how much carbon fixation is due to photosynthesis versus other potential pathways. And finally, the last thing we want to sort of discern with this project is how much primary productivity is being carried out by planktonic microbes, which are those that are, these are floating in the water column, versus the microbes that are making up these mats and microbialites on the bottom of the lake. And so the way we do this is we set up, a set of microcosm experiments where we just use lake water. And then we have another set where we use filtered lake water. So we filter out any planktonic microorganisms, and we add samples of this microbial mat from the bottom of the lake. And so these microcosms will give us a sense of, how much photosynthesis is being carried out by the mats on the bottom of the lake, independent of all the bacteria that may be floating in the water column. So as you can see here on this diagram I've been putting up, this kind of escalates in complexity pretty quickly once you start adding experimental conditions. And so we end up having, about 48 microcosms, I think, per sample site. So the last step in this is we need to spike them all with 14C labeled CO2. We can't do that out in the field or right next to the lake, which would be convenient. So we take all these microcosms on ice back to our lab, and we spike them with a known quantity of radioactive CO2. And once we do that, we incubate them for 30 minutes out in the sun, and we use a water bath that we calibrate to the temperature of the lake water that we measured on sampling day. This usually happens, either, well, it's been the day after we take our samples typically. So we drive down to the lake. We'll collect all of our samples, we'll drive back to Bozeman, we'll get back home around 10 to midnight, wake up the next day, go to the lab and add our 14C, and we'll set out these water baths for half an hour in the sun. to let these microbes, do their thing and fix as much carbon as they're going to. We'll then flash freeze them on dry ice or in a -80-degree freezer to kill all the cells, halt all biological activity, and then we can process them sort of at our leisure. So the way we do that is we add acid to drive off any CO2 that's still dissolved in the water. Then we'll filter out the cells and we'll wash them a few times with pure water in the lab to wash out any dissolved, if they make waste products that this 14C has gotten into, we'll wash those off. And then what remains on our filter is biomass. And we assume that whatever radioactivity is associated with that filter is radioactive carbon that has been assimilated into biomass during the course of the experiment. So the things we can address with this project design are, using our live versus heat killed controls, we can find out what's a biological source of carbon fixation in the lake versus an abiotic sort of just chemical reaction that might result in CO2 precipitating. We can determine what's a photosynthetic carbon fixation reaction versus a light independent microbial metabolism. And we can estimate the primary production of both the planktonic or water column microbes and the benthic or lake floor microbialite microbes. And so I mentioned this a minute ago, but we have these sort of branching, with each variable we want to test, we double the number of microcosms we test, and we typically like to have 3 replicates for each experimental condition so we can get a nice average and an idea of the, the sort of the spread in our data. So this ends up being 24 microcosms per sample site. We sample 2 sites per trip. And we actually have, we do a couple other experiments while we're out here with different carbon sources. So this ends up being a lot of bottles each sampling trip. So here's how it kind of works on a, a geographic level. We go to Great Salt Lake. Our sampling site is here on Antelope Island. And we have 2 sites, like I mentioned. One is up on the north of the island here. This site has a lot of these microbialites, sort of like Melody has shown. I think this was actually a sample site she used in her experiments a couple years ago. And so at this site, we sample the planktonic or water column microbes and the microbialites on the lake floor. Our second site is out here at White Rock Bay. And this site is a lot more, it's flat, it's muddy, and there are no microbialites here. So we use this as a second sampling site for just the planktonic or water column community. So after we go here, we sample, we go back to the lab in Bozeman and we add our 14C, and then it takes a few days to process all these samples. What does the data look like? So what I'm going to show you here on this graph, on the y-axis, we have femtomoles of carbon assimilated per milliliter per hour for this planktonic community. So a femtomole is a tiny, tiny amount, but we're dealing with these little tiny bottles filled with lake water, and they're only incubating for half an hour. So we're just getting a very small snapshot of the amount of carbon that can be assimilated by, a small portion of the lake in a small amount of time. And you're going to see blue bars and orange bars here. The blue bars represent the amount of carbon we see assimilated to biomass in our dark reactions, and the orange bars represent the amount assimilated in our light reactions. So you can see here, this is about what we might expect to see for a photosynthetically dominated community. We can see that these planktonic cyanobacteria are light dependent, they're photosynthesizing, and we see a lot more carbon fixed when we expose them to light than when we don't. There are some variations between our 2 sites, and we can potentially investigate that as our experiment goes on. But really, this basically lines up with what we want to see. We want to see photosynthesis happening, and this is good evidence of it. When we do our microbial mat experiments, one thing I want you to notice right away is that the scale is different on our y-axis. Now we're dealing with picomoles. This is still a tiny amount of carbon, but it's 1,000 times as much carbon as a femtomole. And so what you're going to see, even just looking at this axis without seeing the data, you can see that the microbial mats are on the order of 1,000 times more productive than the water column. And so what you're going to see here, we normalize, this is picomoles carbon for per gram dry weight of microbial mat material. And you can see here again that this is a photosynthetic community. There's a lot more carbon assimilated when it's exposed to light, which is, again, what we would expect. So I've been showing you a lot of, this is femtomoles and picomoles. This is per gram dry weight per hour. These are kind of abstract numbers. And so I wanted to spend the last couple of slides just trying to put this in context for people. And so I'll warn you ahead of time, these are kind of, it's borderline irresponsible to project this far based on how much data we've collected so far. We want to be able to, eventually with this project, we want to create the first estimates of primary productivity on an annual basis for the lake. That's obviously a really complex calculation. And what I've done on these next couple slides is done a really back of the envelope simplified version of the calculations we'll be doing, just to try and give us a ballpark figure for how much carbon is fixed by these photosynthetic communities. That carbon becomes biomass that can be fed up the food web of Great Salt Lake, feeding all of our brine shrimp, brine flies, and the birds. And so I'll dive into it here. As far as the planktonic side of it goes, from our data, we have averages between our two sites of about 160 femtomoles per hour in the dark and almost 1,000 femtomoles carbon per mil per hour in the light. So per a paper from 2005, we get an estimate of the total volume of the south arm of the lake. So all of these calculations, I'm just working with south arm figures since we have not done these experiments in the north arm. And you can see just by the, by the pictures Melody showed, the north arm is a dramatically different environment, and I wouldn't expect the measurements we're getting in this experiment to be applicable to the north arm. So the volume of the south arm is something on the order of 1 × 10⁻16 milliliters. So that's a huge number. I'm sure you can guess. It's sort of, again, hard to wrap your head around. But we'll just bear with me for these calculations, and we'll try to make some sense of it eventually. We can get a very rough estimate for the hours of daylight per year at Great Salt Lake, just based on its latitude and climate. And so we get about 4,300 hours of daylight per year and 4,300, 4,400 hours of darkness per year. And so I'm going to use these hours of daylight with our daylight productivity estimates, hours of darkness with our dark productivity estimates, and make some wildly borderline irresponsible calculations. And we get to carbon fixation rates of about 56 megamoles of carbon per year. So again, this is just a huge number. We're going to have to wait 1 second to make some sense of it, because I want to go into the benthic mat productivity figures here too. So again, we're making some wild extrapolations. We have our average dark productivity now is 400 picomoles of carbon per mil, or not per mil, sorry, that's per gram dry weight sediment per hour. Light productivity is 2,000 picomoles per gram per hour. A 2014 paper estimates that benthic mats cover approximately 700 square kilometers of the South Arm. We have a 2011 publication estimating this is kind of, again, these are wild estimates, but we're looking at chlorophyll levels of approximately 900 milligrams per square meter. And the way we can use these numbers is that our measurements in this experiment, we can correlate the amount of chlorophyll to the amount of dry weight of mat material. And so again, to make a wild extrapolation, we get our measured rates should correlate with annual carbon fixation of about 39 megamoles of carbon per year. Okay, so we have these big numbers. What do they look like? What can we, how can we relate to this? What do we know about the primary productivity in the lake? So our planktonic productivity estimate was 56.7 megamoles per year. Benthic mat productivity was 39 megamoles carbon per year. And the sum of these numbers gives us 95.7 megamoles of carbon per year. How much is 95.7 megamoles? I'm sure you've all been asking this since I first started talking about this. So 95.7 megamoles is about 4,200 metric tons of CO2. Again, this is hard to wrap your head around. 4,200 metric tons is the mass of about 40 blue whales or 700 elephants. 4,200 metric tons of CO2 is about the amount of CO2 released by a car after it's driven 10.4 million miles. Put another way, it's about the amount of carbon fixed or taken up by 70,000 tree seedlings as they grow over a period of 10 years. So this is, it's a massive amount of carbon being, turned into biomass, taken out of, this is a greenhouse gas being taken out of the atmosphere, turned into biomass in the lake every year. And so, I hope that these numbers are sort of something to help you get your head wrapped around this. And what I want to do for this project in the future is, I mentioned we want to get more accurate estimates here. We think these will actually turn out to be low. So all of these calculations are based on data from our July sampling trip. I actually haven't finished processing all of our microcosms from our October or January trips. And we're actually doing a final trip in April. So the idea here is to get productivity estimates for the summer, the fall, the winter, and the spring. And once we have all that data, we will, refine our calculations we've been doing and try to get a more accurate estimate of, this productivity changes throughout the year. And we want to get an estimate of how much productivity is occurring throughout the year in Great Salt Lake. One thing I can tell you that we know is that microbial biomass in the lake tends to be higher in the winter due to the absence of the brine shrimp and the brine flies, which are the primary predators of these cyanobacteria. So that's why I think these estimates I've gotten right now will turn out to be low. I can tell you from our January sampling trip just a few weeks ago, the first indication that we have a lot more microbial biomass in the water in January is that the filters we used to filter the water got clogged up way faster. In July, I was able to get something like 500 mils to a liter through a filter before it just became too hard to filter any more water. And in the winter, I was only able to get between 150 and 200 mils through a filter before it was totally clogged. So yeah, I'm really excited to continue this project. Like I say, the estimates I gave you here are, in scientific terms, these are wildly irresponsible predictions to make because we really haven't constrained all the variables that go into these calculations very well. And so by the time we get publication quality data, I hope that there will be a lot more work going into this, and I hope we'll have a lot more accurate estimate. And ideally, it's going to be a lot higher than what we've seen today here. So with that, I want to thank especially Dr. Boyd, who's my advisor in graduate school. Libby Phones is another member of the lab, actually my girlfriend, who came out on these sampling trips with me. And from the Great Salt Lake Institute, I want to thank Jamie and Bonnie and all of the student assistants who come out on these fieldwork expeditions and make this work possible. And with that, I will turn it back over. Let's see if I can figure